Hello student, how are you? I hope you all are happy, healthy and very fine at home. What did we learn yesterday? Yesterday we have learned about the steric control that stereo or stereo selection. So that type of the thing we have learned yesterday with the help of the example. There we have considered one scheme number, I think 7, that is erythronolidine A, and how it is nicely, what are the segment, how the carbohydrate type symmetry is apparent over there with the how many chirons are there. 2 chirons generally, A and B we are getting, and finally we got the precursor over there. So today we are going to discuss about transfer of chirality. What does it mean? How the transfer of chirality is taking place, that we are going to learn here. Look at here what is happening. A second unique strategy for controlling stereochemistry involve the utilization of oxidizing chirality in a cyclic or acyclic carbohydrate structure to create another center of chirality, usually in beta position via Claisen type or similar rearrangement. Claisen rearrangement, you know that type of the rearrangement may appear here, and by using that approach we can synthesize that type of the natural product over here. And in this manner a useful methodology can be applied for the introduction of one or more carbon substituent in predetermined manner, and thus in stark that is a PGF2 alpha. This is the one of the target molecule we have considered here, and as we know that target molecule means what? The molecule whose synthesis being planned is known as a target molecule, and here we generally particularly keeping the approach is a Chiron approach, and we are how the transfer of chirality is there. Synthesis from D-glucose. Here is a D-glucose. The focus point in terms of synthetic design is the convergence of carbohydrate derived segment containing 2 hydroxyl group and a double bond with C11, C16 of the target. Scheme number 8 is shown over here, and the 2 centrally located hydroxy group in the original addital intermediate are used to create a trans double bond and one unprotected, mainly here is a trans double bond and one unprotected hydroxy group is the Aricher point for a stereospecific Claisen rearrangement to introduce the C8 sutyl bond in the targets. And note again how preferential protection provide an ideal derivative which voluntarily to chemical manipulation. Ireland synthesis of is another example involving transfer of chirality. How this transfer of chirality can be carried out, in this example we are going to learn over here. Here is a here specifically extended annulate. Here we can annulate that one, we can extend that certain part over there. Look at here, there are the 2 groups present. Here is a Y, Z, and here is a X, and now we can join, we can form the new carbon-carbon bond over there, and there is a transfer of a new chirality in the form of this new chiral bond will be formed over here, and then this after forming as a this bond, we will get as a this type of the segment, and this bond can be transferred from there to here, and we will get as a such type of the carbon skeleton. And as we know that polyhydroxy groups over there, and polyhydroxy group, it is one of the characteristic we are trying to find out the precursor of the carbohydrate type symmetry over there, and by elimination of this OH and OH, and we can get here is a double bond, like this trans double bond will be obtained over there just by dehydration or by carrying out some special type of the reaction over there. You will get as this. And now this is a via chain extension. If you collectively collect this one, you will get as a this type of the molecule, and this molecule can be achieved or can be prepared from the starting material, that is a D-glucose, which is a precursor for the synthesis of this. So this is the concept of transfer of chirality, how we can introduce or we can bring the chirality over here. That is a thing we have seen over here. I think with this, let us stop today here. Remaining we will see in the next lecture. Thank you. Have a nice day.